Alright, class. So, we looked at three reasons, right? Why the inside of the cell is so negative. It's negative for three reasons. Number one being the potassium leak channels. Then we have the sodium-potassium pumps by which we lose net positive charge. And then we have the proteins inside which are negatively charged. Okay. Good? All right. Moving on. Now, we did compare a human cell to a battery, right? And we did mention that in a battery, we see there is separation of charge. And we see the same thing across the plasma membrane of a resting cell. Okay, now few terms. And again, do not let these terms bully you to submission. The first term is current. What do we mean by current? Current is nothing but movement of charge. In case of living cells, we don't say current, but we say action potential. So when you hear the term action potential, just know that we have movement of charge. That's action potential. Potential difference is the same as the separation of charge, meaning the difference, right? Inside negative, outside positive. The difference is called potential difference. Resistance is something that prevents a charge from moving. And then conductance is something that allows the charge to move easily. So these are some terms that helps us understand the concept better. I will probably not going to test you on these terms, but it helps us understand concepts. Okay. We also mentioned, now this is a review slide. We also mentioned that in order for these ions or electrolytes to move across the membrane following laws of diffusion, there has to be permeability. A membrane should be permeable to certain molecules or to certain ions in order to achieve equilibrium, right? Okay. And then we define resting membrane potential by separation of electrical and chemical charges across the membrane at rest. That's resting membrane potential. We also mentioned how negative inside of a cell is. It actually depends on which cells we are looking at. Neurons usually measure minus 70 resting membrane potential, whereas muscle cells are even more negative. All right. Now the question is, we understand that in all cells of your body, inside is more negative as compared to outside. However, can all cells of your body change the polarity? Can all cells allow different ions to move across the membrane to change the polarity? The answer is no. There are only few cells in your body that can be excited, meaning that can change their membrane potential, that can be excited to change the membrane potential. And these cells are your muscle cells. We have three types of muscle cells. We'll start with a picture on the right hand side. We do see skeletal muscles on top, followed by cardiac muscle, and at the very bottom we see smooth muscles. All three types of muscle cells are excitable, meaning you can change the polarity of the membrane by passing an action potential or by passing a current. Which other cells are excitable? Well, neurons, of course. Neurons, which we see on the right hand side, on the left hand side, we see different types of neurons. And remember, all types of neurons are excitable cells. Then glands. We have a lot of exocrine glands in the body as shown by the picture at the bottom of the PowerPoint. And remember, glands are very, very excitable. All right. So we do understand that muscles, glands, and neurons are excitable cells and they can change the membrane permeability. But why and how? How can they change and why can they change? Well, these cells have the ability to suddenly and instantaneously change the permeability across their cell membrane. We know that. They have, the reason why is that they have different kinds of gene expression. They express genes that lead to proteins that are integral proteins which span across the membrane, and these proteins create channels, and these channels are gated, meaning these channels can open and close just like we open and close the doors of our houses, and that causes these cells to be excitable. As these channels open, different ions can move either inside or outside, leading to change in membrane potential of these specific cells. All right, now let's talk about these gated channels now. Okay, now before I talk about this slide, let's talk about our houses. If you think of your house, how many gates do you have? Well, you have the front door, then you have your garage door. And sometimes students say, Rupa, I can also enter the house by the sidewalk. Well, let's not think of the sidewalk. Let's think of the front door and the garage door, the two entrances that we use most of the time. Okay. Now, what about the garage door? What about the entrance, the main entrance of your house? Do you need a key to walk in? Yes, you do. You can consider your front door as ligand-gated channels, meaning you need a chemical or a ligand to open the gate. Think of the ligand as a key. So you need a key to open the lock. Or we can say you need a ligand to open the gate. Same concept. There will be binding of the lock and key. And we can think there will be binding of the ligand to its receptor to open the door. On the left hand side, why am I getting confused between left and right? Anyway, on the right hand side top, you do see ligand-gated ion channels in blue. Now you do see the pink Y-shaped molecules here. These are receptor proteins. And do you see the green square things? These are the ligands. The idea is the ligand physically binds to the receptor, and binding of the ligand to the receptor opens up the channel, and the ions can flow in. The blue balls represent the ions. I want you to understand, as the ligand binds to the receptor, the ligand doesn't come in, right? The green balls doesn't come in, but the ligand simply works as a key to open up the gate so that the ions can move in. These are ligand-gated channels. The ligand-gated channels that we will talk about today, well, the ligand actually that we will mainly focus on today is acetylcholine. We will also talk a little bit about norepinephrine as well. Okay, so acetylcholine is a ligand. Okay, other ligands include norepinephrine, epinephrine, dopamine, the catecholamines, right? Those are also ligands. And again, we will talk about those later. Okay, so that was your front door. How about your garage door? What do you need to open your garage door? Think. I want you to think about this. So, in order to open your garage door, you need some sort of transponder, right? So with the little transponder thingy, which is in your car, you can open actually the garage door maybe 100, 200 feet away from the garage. So there is this electrical signaling. Do you agree? There is this electrical signaling from the transponder, from the little clicker to the transponder, so it can help opening up the garage door. So the garage door is opening, but in response to some current, to some electrical event. So those type of gates are called voltage gated. So think your garage door. Those are called voltage-gated ion channels. And remember, they always need some current or some action potential to be opened. Voltage-gated ion channels, they need current to be opened. In the diagram at the bottom, we do see voltage-gated channels, and they open in response to certain change in membrane potential. Now, few things. Let's look at the picture. Voltage-gated channels can have different gating mechanism. For example, if you look here, let me try to change color. Okay, if you look here, this one on the very, very right, you see that the gate is closed. And the gate is closed because, do you see the chain and the ball thingy? The chain and the ball thing, the ball is actually blocking it. So it's called closed. Now when the ball is released, then the gate open up, and that's the open state. However, there is another state where the ball is hanging, but the protein has changed configuration and it will not allow anything else to pass. That is deactivated state. So we have closed state, open state, and deactivated state. I want you to understand that when the gate is closed, nothing can pass through. When the gate is deactivated, nothing can pass through. But only when the gate is open, then molecules can pass through. Let's look at another diagram. Actually, we'll come to this a little later. Okay, good. So we understand closed state, open state, and deactivated state. Very, very important concept. When we talk about action potential, we will come back to this. Please know inactivated, closed, open, and deactivated states of voltage-gated channels. Alright, so let's talk a little bit more about membrane channels. We have ligand-gated channels, remember lock and key for your front door, and then we have voltage-gated channels, your garage door. Okay, so ligand-gated channels obviously need a ligand, and we will talk about two types of ligand, acetylcholine, and then the other ligand we will talk about are norepinephrine or epinephrine. Okay, now acetylcholine is a ligand that will bind to receptor, right? There is binding of the ligand to the receptor, and the receptors to which acetylcholine binds, those are called cholinergic receptors. So acetylcholine binds to cholinergic receptors. Norepinephrine and epinephrine binds to adrenergic receptor. They bind to adrenergic receptor. Now there are two types of cholinergic receptors. How many? Two types of cholinergic receptors. We have something called nicotinic receptor, and then we have muscarinic receptor. I'll make another video to explain these.